Welcome to the Deep Dive. Today, we're jumping into a foundational resource for anyone studying the nervous system, a really comprehensive anatomy and systems atlas.
It really is. And our mission here is to distill it all for you, to give you a structured way to think about it. We're going from a single cell all the way up to the brain's entire protective architecture.
It's all about that organized complexity.
Exactly. The nervous system is nothing if not that. Okay, so let's unpack this.
Let's start at the very bottom. With the neuron, the atlas makes it so clear right from the start that these cells are metabolically, well, they're incredibly frail.
Absolutely. They have basically zero metabolic reserve. They need oxygen and glucose delivered, moment to moment.
Any interruption is catastrophic.
It is. And their shape, their very form, is tied directly to what they do. Those big branching dendrites, that's all about gathering information. And the wild thing is they're not static.
They show plasticity.
Yeah, they can expand or even shrink back depending on what the local environment needs. The whole system is constantly adapting.
But a neuron can't do any of this alone. It needs its support crew, the glial cells.
And what a support crew. The numbers are just staggering. You're looking at roughly 10 times more glial cells than neurons.
Which has huge clinical implications. I mean,
Yeah.
when you think about brain tumors, it's the glia that form them, the gliomas, not the neurons.
Precisely. And within that group, you have the astrocytes. You can think of them as the real workhorses. They form this beautiful interlocked web, a syncytium, that gives physical support.
But it's so much more than that. They're metabolic partners.
Oh, completely. They feed neurons lactate. They help balance ions by soaking up extra potassium. And this is critical, they vacuum up neurotransmitters like glutamate and GABA from the synapse. They're constantly recycling.
And clinically, they're such a double-edged sword. On one hand, their little end feet processes are a huge part of the blood-brain barrier, which is protective.
But after an injury,
After an injury, they form the glial scar. And that scar can become a real problem, either an irritable spot that triggers seizures or...
or it can transform into an invasive astrocytoma.
Exactly. Then you've got the other major player, the microglia.
The brain's own dedicated immune system. They're derived from mesenchyme, and they're basically scavenger cells.
So they're cleaning up debris, pathogens.
And they're also remodeling synapses,
Yeah.
literally pruning connections, which is just fascinating.
And when they get activated, they change shape, right? They become amoeboid and just start pumping out inflammatory molecules.
An inflammatory powerhouse is a great way to put it. They release reactive oxygen species, cytokines like IL-1 and TNF-alpha.
Which is key in neurodegenerative diseases like Alzheimer's.
It's central. The microglia are trying to do their job. They're trying to clear away the amyloid plaques and tau protein, but the response becomes chronic, and that sustained inflammation ends up contributing to the damage.
Okay, so that's the support system.
Yeah.
But how does the neuron itself manage its own internal logistics? I mean, an axon can be a meter long.
That's where axonal transport comes in. It's the cell's internal highway.
Yeah.
You've got fast anterograde transport, which is from the cell body down to the terminal.
Shipping out the necessary supplies.
Yep. Synaptic vesicles, mitochondria, proteins for the membrane.
Wow.
And it moves in this sort of start and stop saltatory way.
And there's a return trip.
There is. Fast retrograde transport. It brings back the used stuff endosomes, damaged organelles, but also growth factors.
But this is also a vulnerability.
A huge one. This is the exact pathway that certain pathogens use to invade the nervous system. The classic example you have to know is the polio virus. It hijacks this retrograde system to get to the motor neurons.
Chilling. And what about regrowth after an injury?
That's where slow anterograde transport comes in. It carries the building blocks, the microtubules, and neurofilaments. But the key insight, the really critical piece of information here, is the speed.
It's incredibly slow.
It's agonizingly slow. It proceeds at about 1 millimeter per day. And that's not just a number for a test. That 1 millimeter a day is the biological reason why recovering from a bad peripheral nerve injury takes so long. You are literally regrowing that axon 1 millimeter at a time.
That cellular view really drives home how fragile everything is. So let's zoom out. How does the body protect this whole system? This is where it gets really interesting for me. The layers, the fluids, the whole architecture.
It's an incredible multi-layered defense. You start with the meninges.
Yeah.
The innermost layer, the pia mater, is almost like shrink wrap on the brain.
Clings to every surface.
Exactly. Then you have the arachnoid membrane, which kind of spans over the contours like a spider web. And the space between them,
The subarachnoid space.
that's where the magic happens. You've got the cerebrospinal fluid, the CSF circulating there, and all the major arteries and veins are traveling through it.
And this anatomy is so directly tied to clinical problems. A berry aneurysm ruptures, and you get a subarachnoid hemorrhage. It's bleeding right into that space.
Precisely. Then, on the outside, you have the dura mater, the tough mother. It's a really durable, protective layer, but trauma can tear the tiny bridging veins that drain into it.
And that leads to a subdural hematoma, which is a slower bleed, right?
Sure, because it's venous.
It's venous, exactly. Contrast that with a skull fracture, one that tears a meningeal artery, that blood pools in the potential space above the dura.
The epidural space. And that's a fast arterial bleed. A true emergency.
Knowing the layers tells you what to expect.
Okay, let's talk about the fluid itself, the CSF. It's like this internal plumbing system.
A vital one. It's produced inside, flows through the ventricles from front to back, and then it gets absorbed back into the venous system through these little one-way valves called arachnoid granulations.
And it provides buoyancy. I remember learning that without CSF, just turning your head too fast could damage the brain.
It would literally shear the long tracts in the brainstem.
Yeah.
But that system has choke points. If the cerebral aqueduct gets blocked, for example, you get internal hydrocephalus. Fluid builds up upstream. And there are congenital issues, too, like with the exit foramina.
Right, the foramina of Luschka and Magendie. If they're blocked, like in Dandy-Walker syndrome, the CSF can't get out into the subarachnoid space at all.
Now, what about the blood supply that's feeding this whole thing?
Two main sources. You've got the paired internal carotid arteries for the anterior circulation and the paired vertebral arteries for the posterior.
And they all meet up in the circle of Willis.
This magnificent ring of 9 interconnected arteries at the base of the brain. It's an amazing piece of engineering, but it's also a weak point. Those junctions are where berry aneurysms love to form.
And for strokes, the key is knowing the territories, right?
Yeah.
ACA, MCA, PCA.
Absolutely. An occlusion in one of those vessels gives you a very predictable set of deficits based on what part of the brain it supplies.
And to see all this, we turn to imaging. Let's start with CT scans.
CT is fast. It's X-ray based, so it's fantastic for bone and for seeing fresh blood. And if you use contrast, you can see tumors because the contrast leaks out where the blood-brain barrier is broken.
But for fine detail, MRI is king.
For soft tissue, absolutely. T1 weighted images are great for looking at the detailed anatomy. On a T1, the CSF and ventricles look dark.
But if you're looking for a problem, you want a T2.
You flip to T2 because on a T2 weighted image, fluid is bright white. So that means edema, an infarct, a tumor, the demyelinating plaques of MRV, they all just light up.
So T1 anatomy, T2 pathology, and now we have MRA and MRV.
Right. Magnetic resonance angiography and venography, they use flow principles to see the vessels without being invasive. They've pretty much replaced the old risky contrast angiograms for most diagnostic work.
Okay, if we take that anatomy now and connect it to the bigger picture, understanding how damage translates into actual symptoms is, that's the whole game in diagnostics.
It is. And the first big distinction you have to make is between a nerve root lesion and a peripheral nerve lesion.
Let's use sensation as the example. Dermatomes, the areas supplied by a single spinal root, are like overlapping shingles on a roof.
That's a perfect analogy. Because of that overlap from the segment above and the segment below, if you knock out one single nerve root, you almost never get complete numbness.
But you do get pain if the root is irritated, like from a herniated disc. That's radiculopathy.
Exactly. That radiating shooting pain right in that principal dermatome. Now contrast that with a peripheral nerve like the radial, median, or ulnar nerve.
Those are the final wires. No overlap.
No overlap at all. So if you cut one of those, you get total anesthesia and complete paralysis in its specific territory. It's all or nothing.
The upper limb gives us all the classic examples. A radial nerve lesion.
You get wrist drop. You can't extend your wrists or fingers.
Median nerve.
Carpal tunnel syndrome is the big one. Weakness in the thumb, wasting of the thenar muscles, and that classic pain and tingling over the first 3.5 fingers.
And the ulnar nerve lesion gives you that really memorable sign.
The claw hand, wasting of the small hand muscles, can't abduct or adduct your fingers. It's a very distinct posture.
And to coordinate all this, the spinal cord is running all these reflexes in the background.
All the time. The simplest one, the one everyone tests, is the muscle stretch reflex.
The knee-jerk reflex.
That's the one. It's monosynaptic. The sensor detects stretch, sends a signal, and immediately activates the motor neuron to contract that same muscle. Super simple, super fast.
But there are more complex ones, protective ones.
Like the Golgi tendon organ reflex. This one is dysynaptic, and it's a high threshold reflex. It kicks in when there's a huge amount of force on a tendon.
And it does the opposite of the stretch reflex, right? It causes inhibition.
It does. It tells the muscle to relax to prevent it from tearing itself off the bone. It's a safety override.
And you mentioned Renshaw cells.
Yeah, they provide what's called recurrent inhibition. It's a feedback loop. A motor neuron fires, and it also activates a Renshaw cell, which then comes back and inhibits that same motor neuron pool. It's a way of fine-tuning and controlling the output.
So we've gone from the cell, through the wiring, up the spinal cord. What does this all mean for the big picture stuff? For behavior, memory, emotion? That brings us to the higher order systems, like the limbic system.
The limbic system is really a functional concept. It's a ring, a limbus, of structures that handle emotional responses, motivation, memory.
The hippocampus and amygdala are the big players.
They're the superstars. The hippocampus is absolutely essential for consolidating short-term memories into long-term ones. And it's a fragile area. The CA1 neurons in particular are very vulnerable to ischemia.
Even a brief loss of oxygen can cause damage there.
It can. Then you have the amygdala, which is all about adding emotional color to our experiences, especially fear. If you have bilateral damage to the amygdala, you effectively abolish the fear response.
Shifting gears a bit, let's talk about the big neuromodulatory systems, the dopaminergic pathways. There are two main ones to know.
The two great systems, yeah. First is the nigrostriatal pathway. It runs from the substantia nigra to the striatum, and it's all about movement.
So when that degenerates, you get Parkinson's disease.
That's the direct cause of Parkinson's. The loss of those dopamine neurons leads to the classic resting tremor, rigidity, and that profound slowness of movement, the bradykinesia.
And the drugs like levodopa are trying to boost dopamine in that specific system.
Exactly. Now, the second system is the mesolimbic and mesocortical pathway. This one starts in the ventral tegmental area, the VTA, and projects to the nucleus accumbens and the frontal cortex.
And this one is about what? Reward. Motivation.
All of the above. Reward, motivation, addiction, attention, cognition. It's a huge player in our executive function and drive. And because of that, it's the target for antipsychotic drugs used in schizophrenia or stimulants for ADHD.
Okay, last one. The cholinergic pathways.
These neurons mostly live in the basal forebrain in a spot called the nucleus basalis of minor. They project everywhere, all over the cortex and to the hippocampus.
And they're tied to memory.
Strongly tied to memory and cortical activation, just sort of keeping the cortex awake. This is why their degeneration is a major feature of Alzheimer's disease. The drugs we use for Alzheimer's are often aimed at boosting this cholinergic system.
Let's try to pull this all together and close out with a few really high-yield clinical syndromes that connect these specific brain regions to symptoms.
Good idea. We can start with just the basic autonomic control of the heart. Sympathetic nerves, speed it up, parasympathetic nerves. Via the vagus, slow it down.
Which means if you damage the vagus nerve, you can get a sustained high heart rate, tachycardia, because the sympathetic system is just running unopposed.
Absolutely. And on the flip side, too much vagal activity can cause bradycardia or even fibrillation.
The adrenal gland is another fascinating example.
It's so unique. It's innervated directly by preganglionic sympathetic fibers. They don't stop at a ganglion. They go straight to the adrenal medulla.
And that direct signal causes it to dump adrenaline and noradrenaline right into the bloodstream.
Exactly. 80% epinephrine, 20% norepinephrine. It creates that massive body-wide fight-or-flight surge.
And you see this balance everywhere. Like in male reproductive function, you need parasympathetic for erection, which is vascular, and sympathetic for ejaculation, which is muscular contraction.
A perfect example of that balance. Now, for the brainstem vascular syndromes, because everything is packed so tightly in there, a small stroke can cause devastating but very specific deficits.
Like Weber's syndrome.
Weber's is a medial midbrain lesion. You get contralateral hemiplegia paralysis on the opposite side of the body, but you also get an ipsilateral, same-sided, fixed, and dilated pupil because you've hit the oculomotor nerve, CN the third, right as it's coming out.
Okay, so contrast that with something more lateral, like the lateral pontine syndrome.
That's usually an AICA occlusion. And because it's lateral, you hit different tracts, you get this complex mix. Ipsilateral facial sensory loss and limitaxia, but contralateral body sensory loss. The anatomy predicts the symptoms perfectly.
And the final catastrophic event is herniation.
When a mass pushes the brain down through the tentorium, you get this cascade of signs. Loss of consciousness, that awful decerebrate posturing with extensor rigidity.
And you lose the pupillary light reflexor.
Because you're physically compressing cranial nerve the third against the edge of the tentorium. It's a dire sign.
Wow. Okay, that brings us to the end of our deep dive. We've gone all the way from the microglia, cleaning up cellular debris, to these massive system-level syndromes.
And that's the foundation. Understanding the structure, the why, is the only way to understand the symptoms, the how.
And that raises an important question for you, the learner, to take away. Let's do a little review exercise. Imagine a patient comes in with that characteristic claw hand posture. They also tell you they have trouble feeling a pinprick on the palm of their little finger.
Okay.
So based on that specific pattern of motor and sensory loss, which single peripheral nerve is most likely affected? And, just as important, which other major nerve of the hand is definitely spared?
Think about those specific territories we discussed. That direct link between anatomy and function is the key to all of this.
Thank you for joining us on this deep dive. We really hope this gives you a solid, organized framework for your ongoing study of the nervous system.